Well, hello everybody. After distillation and absorption, today we are going to discuss the process of adsorption. As you all know, the basics were discussed during the introduction part, and you know that in adsorption, what do we have? We have the mass transfer in this case either between a gas and a solid or between a liquid and a solid. So, what we do, the gas or the liquid, for example, the fluid, it is brought in intimate contact with the solid, and then the components, there are some components on which the solids have got a preferential affinity. So, those components, they are more occluded on the surface of the solid, and as a result of this preferential affinity towards some particular components of the fluid mixture, we get some sort of an enrichment or a separation or a purification. Now, it was already discussed and we will be referring to these two terms during our entire discussion, that is the adsorbent. Adsorbent is the solid on which the adsorption occurs. Quite naturally, we know that adsorption is a surface phenomena. So, if you have a larger surface area, then definitely adsorption is going to be better, and therefore, often we prefer a porous material of a very high specific surface area, and the components which get occluded on the solid surface, they are referred to as the adsorbate. Right. Now, normally what we find is that when adsorption occurs, there are different mechanisms by which the adsorption can occur. Generally, we find that the adsorption, they have got a large number of applications, and primarily they are used for removal of color, odor or some sort of an objectionable taste from say food materials, or maybe from some sort of maybe separation of paraffinic and aromatic hydrocarbons. And a very important application for gas adsorption is your drying or dehumidifying of gases, or maybe recovery of some particular very valuable solvent vapor from dilute gaseous mixture. Quite naturally, we will go for this particular process when the relative volatilities are very less, or maybe when there is no suitable solvent in order to perform absorption or stripping. Under these conditions, we usually go for these processes. And as I have already mentioned, in all these applications, the mixture which has from which the components have to be separated, say we have a mixture of A plus B, and we would like to separate B. We have some particular solid adsorbent which has a greater affinity of towards B as compared to A. So, naturally, when this particular mixture, it is thoroughly mixed or it is brought in contact with this solid, then this solid, it preferentially adsorbs B, and as a result of this particular adsorption, we get some sort of a separation between A and B. Now, how much it is going to be adsorbed? Again, in this particular case, that is going to be governed by the equilibrium relationships, and you would be remembering that the equilibrium relationships in this particular case, it is quite naturally, it will be between the amount of adsorbate in the adsorbent, which is usually shown as Q, and the concentration or the partial pressure of the adsorbate in the fluid mixture, right. Generally, when it is a liquid solution, we express the composition in terms of concentration. When it is a gaseous mixture, often in place of concentration, we prefer the partial pressure. So, it can always be that it is C by P as a function of Q, or maybe Q as a function of C or P. In any particular way, we can express it. And we have to remember adsorption in this case also, it is governed by the equilibrium adsorption isotherm, which was discussed during our equilibrium during our discussions on phase equilibrium. And the best or the maximum possible adsorption that can occur, that is again governed by the phase equilibrium considerations, or the equilibrium between the adsorbate concentration in the solid as well as in the fluid. So, therefore, this is also a mass transfer, interphase mass transfer, which is governed by equilibrium considerations. Now, how does adsorption take place? I understand there has been some basics of adsorption taught to you in your mass transfer class. So, we know that there are primarily two ways by which the adsorbate is occluded or held to the surface of the adsorbent. There can either be a physical bond between them, a weak physical bond like a van der Waals forces, etc. There can also be a chemical bonding between them. Quite naturally, if there is a chemical bonding, it is going to be stronger, it is going to be irreversible, it will be associated with a higher enthalpy of adsorption. It's very natural. And when it is physisorption or when the bond is much weaker, we will be having a lower enthalpy, it is going to be reversible, and under this case normally we get a multi-layer adsorption, while in the case of chemisorption we get a monolayer adsorption. Now, apart from these two mechanisms, there is also a third mechanism. The third mechanism is known as surface condensation. Any idea under what condition this takes place? What does it mean? Now, during surface condensation, what happens? This takes place for gas phase adsorption, and surface condensation, this is observed only for porous adsorbents. We do not get surface condensation for non-porous adsorbents, and even for adsorbents where the pore size is extremely narrow on the molecular scale. Under that condition also we do not get it. Now, even for a porous adsorbent, for when there is a very low relative vapor pressure, or in other words when the partial pressure by the vapor pressure, when this is very low, then for a porous adsorbent where the capillaries are not too narrow on the molecular scale, under that condition more or less the adsorption mechanism is the same as that for a non-porous adsorbent. That is, adsorption occurs by physisorption or chemisorption, as the case may be. As we increase the pressure, what happens? Moment we go to increase the pressure, under that condition we find that the adsorbate, when it is adsorbed on the solid and then it goes into the pores, and inside the pores the condensation occurs. Now, and when the condensation occurs inside the pores, quite naturally the heat of adsorption becomes similar to the heat of condensation. And in this particular process, it is known as the capillary condensation. Can you tell me why this occurs? This primarily, and now remember one thing, this occurs even when the temperature of the bulk fluid, it is above the dew point temperature. So, when the bulk fluid temperature, it is above the dew point, so there is no chances of condensation in the gas phase, but when this particular gas, it is occluded inside the pores, what happens? Moment it goes inside the pores, we find that there is a curved meniscus inside of the liquid which is condensing inside the pores. And it is very important for you to remember that the vapor pressure over the curved surface, it is different as compared to the vapor pressure over a planar surface. More importantly, the vapor pressure over a concave surface we find that the vapor pressure, it is less as compared to the vapor pressure over a plane or a convex surface. And inside the pores, we find that the vapor pressure, inside the pores we find that the liquid meniscus, it is actually concave. As a result, we find that the vapor pressure inside the pore, it is less as compared to the vapor pressure in the bulk fluid. This results in the condensation of the vapor at a pressure lower than the vapor pressure of the same gas over a flat surface. And due to this lowering of the vapor pressure, due to the curvature of the liquid surface inside the pore, capillary condensation occurs. There is another very interesting phenomena associated with capillary condensation. This is also associated with the difference in the liquid meniscus shape during two particular processes, that is during adsorption and during desorption. Adsorption means that when we start from a very low concentration and then we keep on increasing the amount of the equilibrium partial pressure, and accordingly the amount of adsorption on the adsorbent, that part also increases, and we keep on going for higher and higher partial pressures. Now, if we repeat the same experiment and try to generate the equilibrium data by lowering the partial pressure and the Q correspondingly, or in other words, if we perform the process of desorption, then we find that the equilibrium curve which is followed during desorption is different as compared to the equilibrium curve followed during adsorption, and the primary, and every time you will find that the desorption pressure will be always lower than the corresponding adsorption pressure, as has been shown in this particular equilibrium curve. This can also be attributed to the difference in the shape of the meniscus. Usually during adsorption we find the meniscus are either cylindrical or spherical, while during desorption the meniscus are always spherical. As a result, the equilibrium curve generated during adsorption is different as compared to the equilibrium curve generated during desorption, and therefore, whenever gas phase adsorption occurs in porous medium, when the pores are not very small in the molecular scale and the pressure is more or less moderate, we find the phenomena of hysteresis. This is not exhibited during liquid phase adsorption, as capillary condensation does not occur during liquid phase adsorption, as a result of which we find that liquid phase adsorptions, they are more or less reversible. Yeah. Well, for every mass transfer operations, we have found that the equilibrium relationships are very important, because they show that the maximum amount of mass transfer that occurs, right. And definitely in this particular case, as I have shown you, that if you are plotting Q versus C, or in other words, if you are plotting the adsorbate loading capacity as a function of adsorbate concentration in the process stream, generally these are plotted at a given temperature, and therefore they are known as isotherms. And these graphs are very important, because depending upon the isotherms, we know that for any particular adsorption process, suppose we would like to reduce the concentration from say one particular C to the other particular C1, then from this particular graph, once we generate, we know what will be the amount of Q that will be required, or rather what is the Q that we have to provide, and from the Q we can find out what is the adsorption capacity, or rather how much amount of adsorbent should be provided for any particular adsorption operation. And definitely, since the adsorbent is the primary cost, so that governs the capital cost to a significant extent. So, therefore, the first thing, like all mass transfer operations, we have to generate the adsorption equilibrium or the adsorption isotherms. We have already discussed the different equations describing the isotherms and also the different curves, etc. And so, more or less, just to recall, what are the main factors which influence the adsorption isotherm? Quite naturally, it is going to be influenced by the nature of the adsorbent. This is true for any particular mass transfer operation. It is also going to depend upon the temperature and relative humidity of the vapor or the gas stream. or also for the liquid stream also it is going to depend upon the temperature of the liquid. The other thing is, let us see the adsorbent, how it has been prepared, what is the source of the adsorbent. In fact, those things also govern the structure, the molecular structure, the porosity of the adsorbent. So, therefore, this is also going to depend upon the origin and the method of preparation of the adsorbent. For gas phase, definitely you have come to know that it is going to depend upon the hysteresis in the gas phase. And for liquid phases, the liquid has to diffuse into the adsorbent. So, the diffusive characteristics of the liquid are also going to influence this adsorption isotherm, and we know that the diffusive characteristics of the liquid, they also depend on a large number of factors like temperature, etc. There is one other very important thing on which this adsorption, the equilibrium curve, it depends. Now, when you have an adsorbent, and in that adsorbent, when your the adsorbate or the solute, it is being adsorbed, what happens? Naturally, there is more and more solute which is being adsorbed, again more and more solute will be adsorbed. This continues till we reach the equilibrium concentration. Once we reach the equilibrium concentration, then no more adsorption is possible in this particular case. Now, under that condition, what do? Do you just take the adsorbent and to dispose it off? Yes, under certain conditions, definitely we dispose it off. Under what conditions do we perform the adsorption operation and we simply dispose it off? Definitely when the adsorbent is much more low cost, under that particular condition we would definitely do this particular situation. The other thing is when the regeneration method, if we think of something when it is much more difficult, under that condition we would go for it. It can happen that the non-adsorbed component, that is of a much higher value. So, therefore, that is the desired product and that is much more expensive as compared to the adsorbent. So, therefore, we do not bother about the adsorbent, we simply remove the undesirable components and we dispose it off. We start working with the non-adsorbed component that we have done. It can also happen that chemisorption is much more stronger, as a result of which reversibility is difficult. So, under such particular conditions, what we do? We dispose it. But this is definitely not an economic proposition, and also there are environmental aspects of disposing this used up adsorbent. So, definitely for most of the cases we do not go for disposing, we go for regeneration, and we would be regeneration is nothing but desorbing the components which has been adsorbed from the solid. So, we will be definitely discussing, since this desorption or regeneration is so very important, we are going to discuss about the desorption mechanisms. For the moment, it is important for you to remember that whenever we go for this particular regeneration process, with this repeated adsorption and desorption, naturally what happens? There is some loss of the active surface area, and therefore, there is a gradual reduction in the adsorbent capacity during the entire working life of the adsorbent. Quite naturally, why it happens? It happens due to the chemical contamination or the fouling, which is usually considered to be an irreversible process. There is also something like the hydrothermal deterioration, where due to water exposure at the regeneration temperature, the pore structure, it is sort of a collapsed pore structure occurs. It is thought that this is reversible, but in practice it is not completely reversible. So, naturally with every cycle of adsorption desorption, there is some degeneration of the adsorbent capacity, and therefore, it is also very important that the adsorbent that you are using, whether it is fresh or whether it has undergone different cycles of adsorption and desorption, with every cycle the characteristics are going to change because of the progressive changes in the pore structure. So, therefore, it is quite evident that if you have generated the isotherm, say for fresh activated charcoal, this is not going to hold after the charcoal has gone through say 3 or 4 cycles of adsorption and desorption. Why I am telling you so much about the factors which are influencing the adsorption equilibrium? Just to emphasize upon you that due to this, unlike solubility or vapor liquid equilibrium data, there is very less published reliable data that you can use for design of adsorbers. Even if there are some data maybe on gas phase adsorption, on fresh adsorbents, in liquid phase adsorption there is still lesser data. So, therefore, whenever you have to go for the design of industrial adsorbers, usually what is the conventional practice that is done? Conventional practices, just before the design, experiments are performed in pilot scale in order to develop the adsorption isotherms with the adsorbent which will be used along with the adsorbate which has to be adsorbed under the conditions which are close or which mimic the actual process conditions. So, under the actual process conditions with the actual feed stream that we are going to use and the adsorbent that we are going to use, experiments are performed in order to develop these isotherms, and then from the experimental data we generate the isotherms, and these are used for the design of adsorber beds. So, therefore, more or less you have understood what is adsorption, what are the mechanisms of adsorption, and since this is a mass transfer operation. So, therefore, the adsorption equilibrium, what are the important parts, those things you have understood. Now, just like all mass transfer operations, the two phases they have to be brought in contact, and then the mass transfer occurs. And just like the other mass transfer operations, adsorption operation can also be continuous, but here we have another unique feature, we can have a continuous mode of operation in the semi-continuous mode of operation. In the semi-continuous mode, what you have? You have a packed bed. Through this particular packed bed, the fluid it is introduced through this particular packed bed, right. It goes through the packing, the packing is stationary, it does not move, the fluid moves through the packing, and then the fluid depleted from the adsorbate that comes out from here. So, in this case, since it is only the fluid moving, the solid is in a form of a packed bed, it is stationary. Therefore, this is known as a semi-continuous mode of operation, although the contact is continuous contact, but it is a semi-continuous mode of operation. These are generally, these take place in packed bed, they are known as packed bed adsorption. Packed bed adsorption is the oldest and the most conventional method of adsorption. We will be discussing packed bed adsorption in details, but it is also important for you to remember that continuous mode of operation are gaining increasing importance in the modern times, particularly simulated moving beds, where the Molex and the Parex, these two are the recently developed simulators which are widely used for simulating moving beds. We also have fluidized beds and we have rotary beds. We will not be discussing these operations in details. We will primarily be concentrating our attention on fixed bed adsorption. So, as I have told you that in the fixed bed adsorption, more or less we have a packed bed adsorption, and usually we take up vertical cylinders for this particular case, and in this particular when we take up the vertical cylinder, usually it ensures the symmetric fluid distribution, and particularly when the flow rate is less than 1.2 meter cube per second, we opt for this vertical cylindrical vessels, where the fluid flow is also in a vertical direction. Normally, when we have a gas phase adsorption, for a gas phase or a vapor phase adsorption, the fluid flow occurs in the upflow direction, and when we have a liquid phase adsorption, then under that condition the fluid flow occurs in the downflow mode. And naturally, for something which is very important is that we would like to avoid channeling. This is quite known to you. So, therefore, just to ensure the proper flow distribution, adequate plenum spaces are left above and below the packed bed, or else some perforated baffle plates etc. are used. Inlet and outlet nozzles are placed symmetrically in order to ensure that flow distribution is proper and channeling is minimized. This is something very important because we would like to utilize the entire bed properly. And definitely, when the adsorption operation is occurring, we would not like the adsorbent to be taken away with the gas phase or to be carried away with the liquid phase. So, the bed, it is usually supported by several grids, and over the grid the screens are placed. So, therefore, we can have in a layer sort of arrangement where for each upper layer quite naturally the opening will be progressively smaller. So, when we have the grid and the screen arrangement, and the screens have got progressively lower and lower openings as we go to upper and upper layers, what it does? It prevents the passage of the adsorbent, right. So, therefore, as we go for higher and higher screens, the screens are fitted with smaller openings, and as we go down, we find that the screen of each lower layer has got a higher mechanical strength, quite naturally because it has to support a greater weight of the adsorbent. Usually, what we have over and above this particular adsorbent, we have some larger size particles, usually say ceramic particles or something. It not only keeps the bed in place, but through these ceramic particles etc., the pressure drop. Definitely, when we have larger particles, the interfacial area decreases, but along with that the pressure drop is also less. We can also have a graded system of particles where they are arranged from a larger to a smaller size. For example, for water treatment, what we can do, at the bottom we can use filter, we can use at the bottom we can use some filter blocks, and above the filter blocks we might have your ceramic or gravel particles, and above this we can have sand particles. So, therefore, we are having 3 types of adsorbent in this particular case, and for such a case they are arranged in order of the size, the larger size particles at the bottom and then with progressively smaller and smaller sizes. This we adopt when we take up multiple adsorbent for adsorption. But when we have a single type of adsorbent, generally the adsorbent, it is placed between 2 layers of inert material of larger sizes to keep the bed in position such that and also to allow for some amount of bed expansion when the gas phase is flowing, but the expansion should not be so much that the particles they are moving out from the bed. So, in this particular way the packed bed adsorption is arranged. Now, when we are discussing about the packed bed, there is something very important that you have to think. What is that? We find that whenever suppose the liquid phase is entering, what happens? The liquid phase, it has got some particular initial concentration of the solute here. When the liquid phase, say this is a packed bed, when the liquid phase is entering from here, it has a concentration of say Ci. So, what happens? This is a packed bed. So, initially when it comes in contact with the initial layer of the adsorbent bed, and mass transfer occurs here, and after this the mass transfer occurs within a very short layer, and within this layer the concentration, it drops from the initial concentration to the equilibrium concentration, which will be definitely very less, right. So, therefore, after this, the fluid which is flowing after this, for that fluid we find it is relatively free of the adsorbed component. So, therefore, we find that the fluid which is flowing from there, the material which has to be adsorbed, that gets adsorbed within a thin layer at the beginning, and after that the fluid relatively free from the adsorbate, it flows down and it exits the adsorber column. So, therefore, the effluent that comes out, it is relatively free of the adsorbed substance, correct. Now, after some time what happens? After some time we find that this particular zone where adsorption was occurring, that zone gets saturated. That means, the entire zone has reached an equilibrium concentration of Ce and no mass transfer occurs in this particular zone. Or in other words, this zone, it becomes an equilibrium zone. And then what happens? Mass transfer starts occurring over a zone just below this particular zone. Now, we find that the mass transfer, it occurs in a zone which is just below the equilibrium zone, and here mass transfer occurs. In this particular case, the concentration falls from near Ci to Ce, and after that, of course, the effluent more or less free of the adsorbed material, it flows out. So, therefore, as long as this mass transfer zone, it is there, it is quite away from the exit section, we find that subsequently as time proceeds, this mass transfer zone, it gradually travels downwards. As it travels downwards, it leaves behind an equilibrium zone where no mass transfer is occurring, or that zone, it is not available for mass transfer, and this particular mass transfer zone, MTZ as it is called, this is followed by a zone where mass transfer does not occur, but it is ready for mass transfer. It is known as the active zone, right. And the effluent which comes out, that has got a very low concentration of the adsorbent. So, at any particular point of time or any particular instant of time, or maybe at any particular axial location, what do we find? At any particular axial location, we find that initially we have the mass transfer taking place. Mass transfer takes place as long as the concentration difference is away from the equilibrium considerations. This goes on as long as equilibrium is not reached. Moment equilibrium is reached, we find that mass transfer no longer occurs there, and the mass transfer zone has shifted to slightly lower, or rather slightly after this particular portion. So, at any instant of time, if you look at the adsorber bed, you find that the bed comprises of 3 zones. What are the 3 zones? There will be an equilibrium zone where mass transfer has already occurred. It is no more available for mass transfer, and mass transfer will be occurring at a zone just after the equilibrium zone. This is known as the mass transfer zone. Beyond the mass transfer zone, there will be an active zone where mass transfer has not started, but it is capable of mass transfer. So, with this, I will be ending this particular lecture. We will be starting up with this particular unsteady state operation of the packed bed adsorption, and we will be discussing the different concepts like the breakthrough point, the bed saturation, bed exhaustion, etc. And then we will be discussing the details of a packed bed adsorber. Thank you very much.